Dr. Stephanie Archer completed her PhD at North Carolina State University, where she worked in the Lehman Lab studying how sponges alter community composition and ecosystem function in tropical and subtropical nearshore marine ecosystems. She then moved on to work for Fisheries and Oceans Canada in multiple capacities. There, she studied species interactions and ecosystem function of glass sponge reefs and worked to develop efficient monitoring methods grounded in strong understanding of the behavior and ecology of keystone species. After that, she has been an assistant professor at LUMCON, or the Louisiana Universities Marine Consortium, for 6 years, where she has a lab focused on ecology and conservation of coastal sponge grounds, seagrass beds, oyster reefs, and other biogenic habitats. And so with that, please join me in welcoming Dr. Archer.
Yes, thank you very much for having me. I'm really excited to be here today to talk to you guys about context-dependent effects of simulated grazing on seagrass ecosystems. And we're actually going to move down the coast a little bit over to St. Joe and St. Andrew Bay, but I think the issues there are relevant to the Nature Coast as well. So thank you again for inviting me. Before I get going, I want to say that this project took a village, and there were so many people who helped in so many capacities and so many uh, organizations that provided funding and support for this work. So thank you so much to all of you, and we couldn't have done it without you. The other thing I'm going to say before I jump into it, this is still very much a work in progress. We are still waiting on some analyses to get back to us that might help clarify this picture even further. So if you have thoughts as well as questions, please feel free to share. I'm all ears. All right, so with that, I'll jump in to my introduction. Grazing is a really important function in marine ecosystems. Most of us who work in marine systems are very aware of the examples of how grazers or the lack of grazers can change an ecosystem. So in the Caribbean, we're all pretty familiar with the example of the shift from coral-dominated states to algal-dominated states that coincided with the loss of the diadema and the overfishing of parrotfish. And so this shift was really attributed in large part to the reduction in herbivory on many Caribbean reefs. In the Pacific, Hughes et al. Showed that grazing is really important after a stressor like coral bleaching, where the grazers really help maintain that open space, open hard bottom for new corals to settle and grow. And without the grazers in the system, you get an algal-dominated state and fewer corals settling and fewer corals surviving. In seagrass ecosystems, kind of narrowing into the system we're going to talk about, grazing is a fundamental fundamental process that shapes the ecosystem. Those are the words. The classic example is the trophic cascade involving these mesograzers who are really important for grazing on epiphytes and keeping the seagrass blades clean and allowing the seagrasses to persist and photosynthesize as they need to do. We also have large charismatic grazers in seagrass ecosystems, and a lot of work has gone into examining how their grazing impacts seagrass beds as well. Here you see a figure from Moran and Bjorndal showing that simulated turtle grazing can increase growth in thalassia beds. However, too much of a good thing can be detrimental sometimes. Here, looking at an example from Forkin et al. In Bermuda, where they established cages to exclude turtles from grazing, and they saw a dramatic decline in canopy height and leaf width and leaf length around or outside of these cages where turtles had access. And they actually concluded that turtle grazing may have led to the extermination of some seagrass beds around the Bermuda coast. And of course, many of us who have taken ecology or been trained in ecology are fully aware of the classic example of a trophic cascade, where when otters were removed from many Pacific ecosystems, the urchins bloomed and got out of control and decimated kelp beds. And then as otters were reintroduced and populations thrived, the urchins got under control and kelp has been making a comeback. Urchins also bloom in seagrass ecosystems. These blooms are often periodic. We're still trying to figure out exactly what causes them in many situations. But when they do happen, they can denude large areas of seagrass beds. And this has been reported around Florida, from the Gulf Coast down to Florida Bay, as well as in Australia and the Mediterranean, and really anywhere that you tend to get urchins and seagrasses. In St. Joe Bay, there have been large populations of urchins for quite a while, and these populations did a pretty good job of surviving Hurricane Michael. And since Hurricane Michael, there have been issues with periodic blooms of urchins and causing dieback of the seagrasses in St. Joe Bay, so much so that FWC and the Aquatic Preserve in that region have organized urchin roundups to try to get the public involved in keeping the urchin populations under control. Urchin grazing on seagrasses in St. Joe Bay is actually fairly well studied. Ken Heck and John Valentine did a great job characterizing how the urchins impact seagrasses, and from their work, we actually know that urchin grazing can create persistent bare patches within the seagrass beds. So you're looking at a figure where they used caged experiments to enclose urchins and keep them within a designated area. And the continuous grazing, those triangles in the figure, were high densities of urchins that were kept in the same area for 4 months continuously. And it turns out when you do that, the urchins pretty quickly eat all of the above ground biomass of the seagrass, and 3.5 years later, those patches had not recovered. All right, so all of that led to our questions, which is, are the effects of grazing consistent across space? And does propeller scarring, which is a consistent problem throughout areas where seagrass is found in coastal systems, does that propeller scarring amplify the impact of these urchin grazing events? And so we designed an experiment where we had 12 blocks spread across St. Joe Bay and St. Andrew Bay, 6 blocks in each bay. And within each block, we had 4 plots. These plots were established around existing propeller scars, and we had 2 plots on the propeller scar and 2 plots 5 meters away from the propeller scar. And then we randomly grazed 1 plot on the scar and 1 plot away from the scar. And the grazing that we did was a one-time application of grazing where we used garden shears to clip the seagrass to approximately 0.5 centimeter high. And this is what one of our blocks looked like in St. Joe Bay. So you can hopefully see the propeller scar running in that straight line, straight lighter line. And then this picture was taken immediately after we did the simulated grazing. So those squares of lighter color are our grazing treatment. All right, so this experiment ran just over a year. In April of 2024, we did initial sampling. So we went and collected all of the variables that we were planning to collect throughout the course of the entire experiment before we manipulated the seagrass in any way. After we were done with that initial sampling, which bled into May, we applied the simulated grazing treatments, and then we came back 1 month later in June to sample, and then came again in October and November, came again in February, and then did our final sampling in June of 2025. We measured a whole host of variables, variables around water quality. So that would be water concentrations of ammonia and phosphates and nitrate, nitrite. temperature, salinity, DO, and pH. We also put out loggers to measure light levels and temperature. We measured the percent cover of seagrasses and macroalgae, seagrass shoot density, seagrass canopy height. At the very beginning in that preliminary sample, as well as at the very end of our experiment, we took cores to assess seagrass biomass. We looked at seagrass growth. We also assessed the associated communities through video surveys, visual surveys, and dipnet sampling. And then we took sediment cores to look at organic matter within the sediments. And we also monitored those propeller scars that our experiment was set up around to see if there were any directional changes in scar length and width over the course of our experiment. All right, so throughout the talk, you're going to see this figure up in the corner of my slides. And so this is just a guide to tell you what data I am presenting on in that slide. Not, we don't have all of our data for all of our plots, as you will see later. And also sometimes it's just clearer to only focus in on the grazed plots, for example. But I want you to have a clear picture of what data I am talking about. So this will be up in the corner of the slide. And the data that I am talking about will be circled. So for example, if you saw this up in the corner, I would be presenting data from the grazed plots, both on and off the propeller scar. Whereas if you saw this, it would mean that I am presenting data from the plots that were away from the propeller scar. If nothing is circled, that means I'm showing you data from all plots and all blocks. All right, so the first thing we saw when we came back 1 month later was that grazing had had a lasting effect on the seagrass response variables that we were measuring. And so I'm going to have the plots for the seagrass variables organized in the same way throughout the talk. So I'll spend a little time orienting you to them here. We have the percent change in percent cover of seagrasses. And so this is from the preliminary sampling, percent change from the preliminary sampling, so a positive value means that we gained percent cover, a negative value means we lost percent cover. In all plots, we will follow this, or all slides will follow the same color scheme as well. The no grazing plots will be this pink, whereas the grazing will be this green. So we have percent change in percent cover, percent change in seagrass density, percent change in canopy height of the seagrass, and percent change in growth. And so the take-home message for this slide is when we came back 1 month later, the plots where we had applied the grazing treatment, we had a significant decrease in cover, canopy height, and seagrass growth. We expected that. What we did not expect was to see such a big gradient in the recovery between our blocks. And so what you're looking at on this slide is screenshots of the videos from our fish surveys at our block SJ1 in the preliminary sampling before we did the grazing treatment, and another block SJ6, which was further down the bay. And what we saw when we came back 1 month later was that there had been absolutely no recovery of the above ground biomass in SJ1, whereas when we got out to SJ6, you could not tell that we had applied simulated grazing at all. We know that urchin grazing can create those bare patches, but when Ken Heck was able to get urchin grazing to do this, he kept the urchins there for 4 months, grazing continuously on the same patch of seagrass. In a previous experiment, they kept urchins there for 18 days, and that did not create the persistent bare patches over time. We applied simulated grazing a single time, and so we actually didn't expect to see completely bare sediment when we came back 1 month later. And so, a question that immediately became primary for, in my mind at least, was what is driving this really dramatic difference in recovery? We were wondering if the recovery was going to be the same over space, but we didn't really expect it to be this dramatically different, especially because the difference in recovery that we were seeing was negatively correlated with the what you would consider good scores on those seagrass metrics ahead of time. So sites that had denser seagrass, taller seagrass, seagrass that grew better, were the ones where we were seeing no recovery whatsoever. And so I started to dig into our data to try to figure out what was driving this. And across our seagrass response variables, we saw that they were significantly correlated with sediment organic matter and the percent nitrogen in leaves during that preliminary sampling. And so for people who don't spend a lot of time in seagrass beds or thinking about nitrogen cycling in seagrass beds or nitrogen dynamics in seagrass beds, the reason we measured percent nitrogen in the leaves is because we know that seagrasses can take up extra nutrients that are available in the system and store them in their tissues. And so they can often be a better indicator of kind of longer term nutrient availability at a site than taking a water sample and measuring the nitrogen, the bioavailable nitrogen in that sample. And so these two variables together explained between 19 and 53% of the variance in how seagrasses responded um, in between our preliminary sampling and that first sampling when we went back. When we came back in the fall, it was to find these persistent differences. We still were not getting any recovery at the sites that hadn't recovered within 1 month, and the sites that had recovered within a month still were doing great. And by the time we got to the end of the experiment, the story was much the same. The sites that didn't recover immediately never recovered, but we still had this pretty big gradient in how the plots responded to the simulated grazing. And we do have significant differences in the percent change in our seagrass response variables between our treatments. And the one thing that was interesting to me in this data is that while it does seem like there may be an interaction, a small interaction between propeller scarring and grazing, grazing is definitely the dominant factor here, and the interaction with being on a propeller scar actually isn't statistically significant in any of these models. And so grazing is the bigger driver, and the scar difference was only ever significant in seagrass density. So the other thing we saw over the course of the experiment is that the recovery that we were seeing, or lack of recovery that we were seeing in our experimental plots, was reflected in the seagrass or in the propeller scars that we were conducting our experiment around as well. And so basically the sites where we saw no recovery, those scars actually got worse. They got a little bit longer, a little bit wider over the course of the study, whereas where we saw recovery, those scars almost went away over the course of our year-long experiment. And so this gave me some confidence that what we're seeing in our experimental plots is an actual phenomenon driven by characteristics of the environment and not an experimental artifact that we created by doing something that we didn't realize we were doing differently amongst the blocks. And so after we went back in the fall and saw that those differences were still there and that the blocks that weren't recovering really weren't recovering, we hypothesized that perhaps when we removed that above ground biomass, we removed the oxygen delivery to the sediments, and there had been a buildup of sulfides in the anoxic sediments. So seagrasses do this really cool thing. They photosynthesize above ground, and one of the byproducts of photosynthesis is oxygen. And they actually take that oxygen and they move it down into their roots and rhizomes, and they release it into the sediments and create a small oxic area, a small area of oxygenated sediments around them. And one of the reasons that this is important is because when you get anoxic conditions in the sediments, especially where you have a lot of organic matter, you tend to have sulfide buildup. And sulfide can be a really toxic chemical for a lot of life, including most plants. And so the bacteria that do this process, that turn sulfates into sulfide, need an oxygen-free environment. And so by oxygenating their root area, they reduce the amount of sulfate reducing bacteria and reduce the amount of sulfide and reduce the likelihood that they will encounter toxic conditions. And so we thought that maybe when we removed that above ground biomass, that oxygen delivery stopped, we got sulfide buildup, and that's why we weren't seeing recovery. Okay, so in February and June of 2025, so the winter and summer samplings, we measured pore water sulfide concentrations in situ. So we stuck a rhizome sampler into the sediments and allowed them to do their thing until they had filled up, and we immediately exposed the sample to zinc and killed the bacteria that would continue to create sulfides and measured sulfides in the pore water. We also sent the roots, rhizomes, and leaves taken from the preliminary and final samplings of those cores off for sulfur isotopes. And I will point out that we only did this in the plots away from the scar because measuring sulfides and sulfur isotopes is not something we had originally budgeted for and not something that is extremely cheap to do. So the reason that we wanted to look at sulfur isotopes is that there's a pretty big gradient in the isotopic ratios from seawater to pore water. So seawater typically has positive sulfur isotope values, whereas that's in heavily reduced sediments, you get a really negative sulfur isotope. So if there's a lot of sulfide in the sediments, you tend to get a really negative sulfur isotope. And you can use this to look at intrusion, because if you're getting sulfide intrusion through the sediments, it should show up first in the roots, and then in the rhizomes, and then in the leaves. And so we actually do see evidence for sulfide intrusion, but we kind of see it everywhere. There is this pattern of increasing sulfur isotopes going from a signature more closely related to pore water to more closely related to seawater from the roots to the leaves. So potentially sulfide intrusion, but in the winter, when we looked at the relationship between those pore water sulfide concentrations and our seagrass response variables, we don't actually see anything that is significant. So there are some lines suggesting maybe a trend, but if you notice, those error bars always cross zero. And so there's no significant relationship here. I also want to point out that especially in the winter, the levels of sulfide that we're seeing in the sediments are way below what you would expect to create any problems for thalassia at any point. When we get to the summer, we do start to see trends towards a more significant relationship between pore water sulfides and our seagrass response variables. But the only one that is actually significant, which is the relationship between pore water sulfide and percent change in growth, is actually a positive relationship, which was extremely confusing to me the first time I looked at this data. Um, it does not seem as if sulfide should increase seagrass growth, and I am not suggesting that it does. But this relationship had me scratching my head for quite a while. And when we dug into the sulfide intrusion um, data from the isotopes, we saw that really where the patterns of intrusion and where those plots fall along that isotopic ratio doesn't actually correlate with where we're seeing recovery or not. So this darker line here, that is the block where we saw absolutely no recovery throughout the course of our experiment. And it's actually on the higher side of the isotopes. And so it started to seem like maybe actually sulfide wasn't playing too much of a role. So this is the change in percent cover. Same thing when we look at growth. We're not seeing a relationship between those patterns of sulfide intrusion and patterns of change in seagrass response variables. Also, over the course of our experiment, the sulfur isotopes tended to move closer to seawater, suggesting that sulfides were actually even less of a problem than they were at the beginning of the experiment. Except, I want to point out this darker line right here. So that is the same plot that I pointed out in the last or two slides ago. And so if we pull out just that block, looking at the grazed and ungrazed plots that we measured the isotopes on in this block, we see a different pattern in the change in the sulfur isotope in the roots going from the preliminary to final sampling. So the circles are where we applied the simulated grazing, and the triangles are where we left it alone, our control plot. And so these are going in different directions. And so maybe again, we're trending towards sulfide having something to do with this differential recovery. I do, however, want to point out that -19 for a sulfur isotope is not really that negative. It's not indicative of something where you would expect sulfides to really be a problem. And so this differential kind of response between the cover and the growth and the density and canopy height suggested that maybe we needed to look at this data in a multivariate way. So you're looking at the results of a redundancy analysis. The way to interpret this plot, for those of you who aren't familiar, is just the points that are closer together are more similar. And then the points are colored by the percent change in density, and the size of the points is scaled to the percent change in growth. So the bigger points had a higher change in growth, whereas the really small points actually had a decrease in growth over the course of the experiment. And then I have just percent change in canopy height and percent change in cover overlaid there. So you can see that they trend in the same direction in this plot as the percent change in density. And so what we're seeing is that the grazed plots that lost the most seagrass had the highest growth. These things are actually along different axes in our ordination. And so when we added on our environmental variables that we measured, sulfides and sediment organic matter came out as the important variables explaining about 50% of the variance in this data. But again, it's conditions that promote growth when the grass is undisturbed or when the grass does recolonize. inhibited recovery of things like percent cover and shoot density. And so, what I think is going on is that we have a slightly variable landscape within the sediments in these plots. There likely is a highly reduced atmosphere in a lot of the sediment, but where exactly that those pockets of sulfide are is probably quite variable in kind of the XY axis as well as the depth axis. And so once you do get a shoot that is a seagrass shoot that is able to establish itself, is able to find a pocket of sediment that isn't as reduced, doesn't have as much sulfide, it is able to really take advantage of all of the nutrients and things that seagrasses need to grow that are available in the sediments because the competition has been wiped out. And so the conclusion to me is that sulfides do seem to be playing a role out there, but they aren't the only driver. Again, we've only explained about 50% of the variance in this recovery, and there is something keeping sites with similar levels of organic matter from developing the same amount of sulfides when we applied the simulated grazing. And so that led me into the literature and now hypothesizing that there might be a role for iron in the sediments. So iron can bind really readily with sulfides to produce iron monosulfide, which is not toxic to seagrasses. And so high levels of iron can actually reduce the amount of sulfide that accumulates in the sediments and increase the suitability of those sediments for seagrasses. This idea is not new. I did not come up with it. People are starting to look at iron additions as a recovery and restoration tool. This is an example from the northeastern US where they collected sediments from a bay where there were still seagrasses and a bay where they had lost a lot of seagrass due to sulfide intrusion. And they raised seagrasses in a mesocosm experiment in those sediments, both with and without the addition of iron. And what they found is that the more iron that they were able to get into the sediments, the better the seagrasses survived and the better they grew. And so luckily, we still have sediment sample. So I have just set off those sediment samples for iron content analysis. We're going to look at bulk iron and iron monosulfide content to see if that helps explain why we're seeing differential recovery and why some sites with high organic matter are recovering and others with high organic matter are not. I would be interested, I do not have funding for this at the moment, but I would be interested in going back and doing iron addition experiments to see if we can stimulate recovery at these plots. We also have those videos that we took to look at fishes, and I'd like to see if ray disturbance or bioturbation is also playing a role in maintaining these bare patches. We will eventually, at some point, also be analyzing the associated community to see how this differential recovery of the seagrasses is altering community structure and stability. And so just to wrap up, our question, are there effects of grazing across space? I initially had that saying, are there consistent effects? So yes, there are effects of grazing. Or no, I have it right. Ignore me. No, the effects of grazing are not consistent across space. Conditions that promote seagrass when they're undisturbed tend to hamper recovery when we have disturbance. It does seem to be partially driven by sulfides and sediment organic matter, but other things are playing a role, and it would be really nice to be able to figure those out. About the question with propeller scarring, it does not seem to significantly amplify the impact of grazing, but it is possible that because our experiment covered such a wide gradient of environmental conditions, and those seem to be really driving the recovery overall, that that is more important than the interaction of propeller scarring and grazing. And if you looked at this all within one environmental or more areas of similar environmental conditions that you would see an interaction. And with that, I will go ahead and take any questions or ideas if you have them.
Awesome. Yay. Great job, Stephanie. I love to hear about seagrass stuff. Um, you guys should see that a poll popped up, so if you wouldn't mind filling that out when you get a chance. And then, yeah, we can open this up to questions. Feel free to add them in the chat or unmute yourself, and ask Stephanie. All right, I have a question from Savannah Berry. She says, do you have any noticeable differences in the above to below ground biomass ratios at the sites that did not recover well? In our part of Florida, we have seen that the Valencia, with a lot of leaf relative to below ground, might be less able to weather disturbances, and just curious if that could be playing a role.
Yeah, so we do have quite a range of above to below ground biomass, and um, that actually seems to be quite different between the two bays. And I think the differences between the bays are hampering my ability to see if it is correlated with recovery with across the bays. Um, at the end of the experiment, the only thing we got in the cores from the ones that didn't recover were some roots that had made their way in. Um, so very skewed to below ground in those, but it wasn't anything that ever came out as a consistently strong trend. But that is a trend that they are seeing in salt marshes as well as a diagnostic for when a system is about to collapse. That above to below ground ratio really starts to skew. And so I would definitely believe that that's happening. I think our environmental gradients and the fact that the two bays are really different, and that just statistically means we're not going to pull it out.
Yeah, makes sense. Um, I have another question in the chat from Laura, and she asked, how do sites differ in light and depth? Like that dry differences in recovery too?
So the sites where we saw the best recovery and the sites where we saw the least recovery were really similar in depth. We do have a couple deeper sites, but they tended to be right in the middle. Light was not really very different across them. And um, occasionally it would start to pop out, but as soon as you started to account for things like weather that day that we were measuring and the differences in days where we got the loggers out, it went away. And so I think light is probably not a limiting factor at the sites where we did this experiment, and we're just you could have a different story if you moved deeper or into more tannin-rich waters where, or more eutrophied waters where light is starting to limit things.
Awesome. Anybody else have other questions for Steph? I have another one from Megan Christopher, who said, great presentation. Two questions. Did you consider below ground biomass removal in studying grazing effects as seen with urchin grazing on root and rhizome structure versus above ground grazing by turtles? And did you select scars of similar characteristics, scar depth below ambient grade?
Yeah. So permitting wise, we were allowed to do exactly the type of grazing that we were allowed to do. We had a larger experiment designed, and for a lot of reasons, it got pared down to just the simulated kind of a removal of the above ground biomass by a maybe urchin front moving through. We did not dig into the rhizomes or anything. We left those intact. Though all of our plots were fully connected to the seagrass around them. Um, and yes, our scars were similar in characteristic before we got started. The other thing that we used to select our scars that weren't mentioned was separation from other scars. So we ensured that all of the scars that we used were at least when we set up the experiment, 300 meters from any other scar.
Very cool. I have another one from Caitlin Young, who asked, do you have any idea of the clonality, genetic diversity, or spatial age structure of each block? Curious about the possibility of nutrient translocation and shoot age affecting recovery differences.
I am too, and no, we don't.
I have another question here from Amphis, and they ask, was there continued boat traffic by the scar selection sites?
Yes. These sites are highly trafficked. The sites where we saw no recovery are actually right by the mouth of Eagle Harbor, which is right where the boat rental is for the state park that we worked alongside. And there was the creation of propeller scars closer than 300 meters to many of our sites during the experiment. Um, we actually saw some of them created while we were out there. So yeah, there was definite continued use and continued impacts that we could not control to these systems. But I will say that we more consistently ended up with people right next to us who stayed there and who were using the area out at the sites that did recover. Um, so
Very interesting. I have one other question from Annie Hensel, who asked, was recovered grass all Thalassia, or did you see pioneer species as part of the recovery or macroalgae? I might have missed this early.
yeah, so the differences in macroalgae tended to persist from initial sampling all the way through. Um, and we saw Haladuli move into one plot in one block. And that was a block that already had more Haladuli in it when we established the experiment. So we did not see any insights where there wasn't already significant proportion of the seagrass community as Haladuli. We didn't see any move in.
Interesting. And did you do growth on just Thalassia, or did you do it on
Mhm.
other? Okay,
Yeah, we did. It was
makes
the
sense.
dominant species in
Right.
every plot, for sure.
Yeah, makes sense. Mark has a question. Mark, go ahead.
Hey Steph,
Hey, Mark.
I don't know if this is allowed, but I'm going to do it anyways. I'd like to ask you a question about sponges.
Yeah.
Um,
Happy,
so,
always happy to talk about
well, I
sponges.
knew that you would allow it. So you've worked a ton. I mean, across like Thalassia's range. I was curious if you have some insights on how like sponge diversity varies. Really, I mean, I'm thinking of the latitudes of your, where you've studied over time, and then also like how sponge functional role might be different across throughout Thalassia's range.
Yeah, so for those who don't know me like Mark does, my other work that he's referring to has been in the Bahamas and in Curacao, where there are sponges everywhere in the seagrass. Um, there are sponges in the seagrass. We actually had several blocks where there were consistently sponges that we found. Um, and there are areas of at least St. Joe Bay where there is sponge densities re very equivalent to the Bahamas. That is not where we did the experiment. So, yeah, it's pretty variable across the bays. So I should say because myself and my co-PIs were not familiar with St. Andrew and St. Joe Bay when we started this experiment, we first did a structured survey of as many points as we could get to in both bays. And so that's why I feel confident saying that it's patchy. Um, but yeah, I think the sponge species that we saw in these beds are the same that you see in the Bahamas. Um, and we know that sponges can be really flexible about what they're doing, what they're eating, what nitrogen they're releasing, all that fun stuff, what carbon they're eating, depending on what's around them. So I would be shocked if they're not doing something slightly different because the conditions in these bays is very different than the Bahamas. But unfortunately, I have not convinced anyone to give me money to study that yet. So,
Cool, thanks.
mhm. Maybe
And
Fia can answer it for us in a couple years.
maybe. Any other questions? Laura?
A sort of a similar question, but um, are there, did you look around for like any other sort of organisms that might have some symbiosis with seagrass to help in that recovery? And I'm really thinking sort of those horse mussels that have been looked at in this region.
Yeah, so that we do see a big gradient of in St. Joe Bay.
Mhm.
Um, we don't see a gradient. We see them in St. Andrew Bay, but less of a gradient. They're either there and abundant or they're not at all. Um, so again, I think the differences in the bays is probably hampering what I have pulled out so far. Um, but I haven't specifically looked at the correlation between those and recovery. Anecdotally, I can tell you that we saw them at every site in St. Joe Bay when we did the grazing application.
Mhm.
Um, More than we saw them when we did the visual surveys. And so I think the canopy height is going to be really correlated with how well we sampled those, unfortunately. But they did show up at every site except for the one that didn't recover at all.
Oh, sounds like a fun thing to
Yeah, yeah.
to follow up on.
For sure. For sure. I mean, yeah, anything that increases that kind of exchange between the sediments and or the pore water and the above and the surface water is going to help with that sulfide intrusion, at least to some extent.
Thanks.
Uh, I had a question. Um, about, well, I guess two things. One, like sulfide bacteria
Mhm.
interactions. Have you thought about that?
Yes, in that I asked my co-author Marshall, who
Mhm.
is um, a, a microbiologist, and so he and I have been tossing around ideas, and I have samples collected for him to try to at least get an initial picture of the bacterial profiles in the sediments and associated with the rhizomes and the roots as well across these plots. He just moved his lab from LUMCON to Dauphin Island, and so it'll probably be a little bit before we get those analyzed. But we do have those samples, and I am going to leave it to him to interpret the results.
Awesome.
But yeah, I think it would be logical if there is a difference there.
Yeah, cool. My second question or thought is when you brought up iron into the equation, I was wondering what you think would drive like differences in iron concentrations across this area that you see.
That is a good question. It's not a huge gradient, like spatially. They're not miles and miles apart, right? I mean, the two bays are, but within the bays, I don't know. I haven't spent a lot of time thinking about iron outside of where it would be introduced from an anthropogenic impact, and I can see where that would be different between our two bays. But I don't. Maybe the dredging that they did in Eagle Harbor after Michael might have remineralized some iron. But if that's the case, then you would expect the sites closer to Eagle Harbor to be the ones that had a lot of iron and less sulfides, and that's not totally consistent with what we saw. So I haven't thought about it yet. I'm
No,
hoping someone who knows a lot more about iron than me can help
yeah,
me with that.
totally fair. Totally fair. I was just curious, you know.
Yeah.
I have a comment with this saying some fertilizers have iron as well, and certain water irrigation systems too. So
Yeah.
could
Yep.
also be some inputs of iron.
Yep. For sure. There's some irrigation and landscaping along the plots, but in St. Joe Bay we were along the state park, and in St. Andrew Bay we were along Tyndall, um, the Air Force Base. Sorry for those not familiar with the geography. And so yes, I know that stuff moves around. Groundwater does things, comes up in places you don't expect it to, all of that. And groundwater inputs could definitely be a source of different iron concentrations around, but as far as direct runoff, I'm not sure. Again, different between the bays, but within the bay,
Yeah.
yeah.
Cool. I have another comment from Ryan that says, we recently completed some sediment contamination work in St. Joe Bay that may be of interest that we can send.
Awesome. Thanks, Ryan.
All right. Any other last questions for Stephanie before we wrap this up? All right. Well, Stephanie, thank you so much for joining us and great talk. For everybody, you can please fill out that evaluation poll, and then hopefully we'll see you in a couple weeks for the next talk. Until then, take care, everybody.
Thank you.